Okay. Thank you, Mr. Pfaffel. Good morning to you all. I'm going to show you now a combination of microgenomic approaches to understand the impact of gut microbiota on the regulation of the hypothalamo-pituitary-adrenal axis in rats. Indeed, it's now well acknowledged that gut microbiota can impact behavior, anxiety, and emotivity in lab animals. We too, in our laboratory, have compared the behavior of conventional and germ-free rats in response to stressful conditions. As you can see here, in this panel show that germ-free rats have exhibited more signs of anxiety than conventional, as shown by their diminished visit in the open field and increased number of fecal boli. Serum corticosterone levels were determined, and germ-free rats display higher level than conventional rats in open field stress. So to understand the impact of gut microbiota on the activation and regulation of the HPA axis, we designed an experiment to compare germ-free and conventional rats placed in stressful conditions. Rats were placed 10 minutes in the open field, lit with very bright source. And a multimodal approach was chosen to analyze the result either by studying behavior and brain biochemistry and through metabolomic and proteomic analysis. Here, I will only focus in the transcriptomic analysis. For this, we have selected 48 genes corresponding to the three structures of the axis. Well, in this slide, show three main structures of the HPA axis: hypothalamus with a paraventricular nucleus, hypothalamus releasing CRH, and CRH is acting to corticotropic cells of pituitary, when then secrete ACTH. ACTH then acts on adrenal cortex, specifically cortical zona fasciculata, which finally secretes corticosterone, and corticosterone sends negative feedback to pituitary and hypothalamus. Because of each tissues they are very different, we use a different technique to collect the samples. For hypothalamic PVN, we punch area from 40 micrometer cryostat section and pull 4 of them per sample. For adrenal gland, we use laser capture microdissections to collect zona fasciculata specifically. And for pituitary, we label the cell of interest, corticotropic cells, and also laser capture them. We extract the RNA using different techniques adapted to the amount of material collected. For these two, pico-pure kits were used, and for anterior pituitary gland, we use cell-to-CT kit, which allows to analyze the transcriptome of single cells directly from cell lysate. You can see the integrity of RNA is very good, and retranscription was performed to all RNA. For adrenal and pituitary, we performed preamplification because the amounts collected were too low. The preamplification was carried out using Tachman preamp master mix and custom preamp pool containing all the 48 primers for detection by Tachman GenX expression assays. Relative amplification was validated using single gene by single plex probes for several genes prior to analyze the qPCR. The amplification were homogeneous and about 13 CT, which is equivalent to 1,000-fold increase. We checked that the preamplification was linear for concentration ranging from 3 to 90 nanograms. We use a common quantitative PCR approach to analyze the expression of 48 genes specific to the axis, the microfluidic cards. The microfluidic cards were custom designed for this experiment. And you can see in this slide microfluidic cards with 384 wells. In each wells, there is a couple of primer and Tachman probe for specific one target. Data analysis was performed using the GenX software to validate reference genes with norm finder and genome, to exclude outlier, to calculate delta CT, and to perform statistical analysis appropriate. Very briefly, because the extensive results are still in progress, we already got significant difference in the expression of several genes, either depending on microbiota status or on the stress condition. For conclusion, this approach uses a single set of genes to study their different tissues, different by nature, by heterogeneity. This workflow allows study of all tissues, specific areas, or specific cells, using techniques ranging from classical transcriptomic then to microgenomic. For more results, you can go visit the poster presented by Patricia Anglade. And I want to associate all those involved in this work, Patricia Anglade and Sylvie Rabot from INRA Michaelis Institute, which developed this topic research. Claudia Bévy-Lacroix, transcriptome platform manager in INRA. Partner supplier, Exilon, Pierre Dufresnay and Nicolas Crapard, who participated of the project start. Life Technology, Stephen Jackson, Shirley Shu and Alan Carpino for their advice and supply kit molecular biology and microfluidic cards. And finally, I would like to thank Michael Kubista for lending me the latest version of GenX. Thank you for your attention.
Thank you very much. The talk is open for discussion.
Very interesting observation in the beginning. Could you explain a little bit what is the biological meaning of your finding that germ-free rats have this stress symptoms?
Yes, it's a new topic, and effectively, germ-free rats seems to have very anxiety. compared to conventional rat. And now you try to understand why it is what happened, but we don't know.
Just to add some elements to the answer to your question, because I participated to this work. So there is a growing body of evidence that gut microbiota is participating to the development and functioning of the brain. And there are some groups who have shown that germ-free animals, rats and mice, are more anxious and have a hyperactivity of the HPA axis compared to conventional ones. So the only observation now is that there is a higher level of corticosteroid in the blood, and the idea here was to understand precisely in which structures the HPA axis is dysregulated. That's why we investigated the genes involved in this functioning.
Are germ-free rats completely free of any microbiome? You know, we hear about how the microbiome is so critical for every aspect of life, and here we have a germ-free animal that obviously is pretty good, doing pretty well without any bacteria. Is that true? Or is it just some sets of bacteria that are not present in the
germ-free rat is
completely no microbiome in the rats?
No microbiome.
It's quite significant when we all are believing how the microbiome is so critical to life itself. Lately, in the big excitement about the microbiome. Secondly, since the rats have to be kept in a germ-free situation, wouldn't their environment be different than a rat that is not germ-free? And how do you control for that? I saw in your first couple slides, when you're talking about the free-ranging, is it all the same environment except one is in a sterile environment? Wouldn't there be differences in the containment for a germ-free rat compared to a non-germ-free rat that might affect the environment and that might affect that stress level? You understand my question?
Conventional rats in this study live in the isolator, the same isolator of germ-free rats.
Okay.
To compare in the same condition the rat and the stress.
But you have to sterilize everything you're putting into the cage.
Yes.
So everything is sterilized.
Yes.
Okay, so
The
it's
feed,
otherwise
the
it's
food,
same,
the
it's
straw,
just sterile
everything.
versus non-sterile. Okay, good. Thank you.
One question from my side. Do you have already data about the hormone levels? Do the hormone levels change along the axis? You said the hypothalamus, pituitary, coronal axis. So I think this is the trigger. So do you have already data on the hormonal changes, the concentrations of CRH and ACTH? No. Not yet.
No.
Not yet.
Corticosteroid.
Okay. So thank you very